In Chapter 12, we're going to talk about the eukaryotic pathogens, fungi, algae, protozoa, and helminths, or worms. When we look at the tree, we see that these are going to be more related to animals, the protozoa being usually more simple, and fungi and algae being branches off of that eukaryotic lineage. When we talk about the fungi, we know that they're eukaryotic, so they're going to have a nucleus. We also know that they are aerobic, and some of them are facultative anaerobes. Chemoheterotrophic, and they are the major decomposers within the environment. When we compare the fungi and the bacteria, obviously fungi are eukaryotic, so they will have a nuclei and organelles, but at the cell membranes, they also have sterols present, gives them a little bit more added rigidity to the membrane. They can have cell walls, but not peptidoglycan. They're things like glucans, mannans, and chitins. And if you're looking for a defining feature, it's often the spores. They can produce a variety of spores, both sexual and asexual. And we see that the type of antifungals are not the same as antibiotics used for bacteria. They're often sensitive to things like polyenes, the imidodiazoles, and glucofulin, and these are going to be the common antifungals that you may have already heard about. For the eukaryotes in general, we see that these are going to have a progression from absorptive type of acquisition. So with the fungi, we'll see that their food acquisition is absorptive. As we get up to the helminths, the worms, we're going to start to see a digestive lifestyle. For the fungi, they are the chemoheterotrophs. Many of them are going to be filamentous. Some are fleshy like the mushrooms, but we do have the unicellular yeast as well. When we talk about the nutritional adaptations of fungi, again, as a chemoheterotroph, they're going to absorb their nutrients. They prefer more acidic pH, a pH around 5, and they can grow in a variety of places with not much moisture and or nutrients. So we see that most of these are aerobic, but some of the yeast are facultative anaerobes, and we know if we want the yeast to do good fermentation, we have to put them in an anaerobic environment. In general, they are very resistant to osmotic pressure. That means that they can grow in high salts and high sugars, and so that can be problematic from food standpoint of allowing these to grow even when we have those type of osmotic pressures present. They require less nitrogen than bacteria, and they are the major decomposers in the environment because they are capable of metabolizing very complex carbohydrates that most bacteria cannot. When we talk about the molds, the body is referred to as the thallus. It consists of a lot of hyphal filaments that are going to be thread-like as we look underneath the microscope. A massive hyphae is referred to as a mycelium. When you look at the hyphae, the vegetative hyphae are the ones that are going along the surface. Their job is to obtain nutrients from the environment. Then you will see rising up from the vegetative hyphae an aerial hyphae. At the end of the aerial hyphae is usually a reproductive structure, and so you can see in the picture of the aspergillus that we see the spore structure at the end of the aerial hyphae. In panel B, we see the aspergillus growing on agar. So you can still see the fuzzy nature of the filaments, and you might also notice that they grow in concentric circles. They start in the middle and then keep on sending out those vegetative hyphae in search of food, and so you can actually see the concentric circles as it does so. Out in the environment, you may have noticed a fairy circle where that's like a ring of mushrooms. The same thing is happening. They've started in a center, and as they keep spreading out, you get an expanding ring for the fungi as they grow. Yeast, on the other hand, are unicellular. They can divide by fission, and this can be either symmetrically or asymmetrically. When we think about the budding, that means that it's pinching off just a little bit of a daughter cell. Typically, we see this is happening with pseudohyphae. It's just really an elongation of that unicellular fungi. It's not a true hyphae. And then you can see where the smaller bud is pinching off. In the picture, you also see a mother cell that has had several daughter cells bud off, as noticed by the bud scars. The interesting thing about the fungal pathogens is many of them are dimorphic. This means that they have two different morphologies, and this is controlled by temperature. At 37 degrees, which is body temperature, they're usually yeast-like. At 25 degrees Celsius, which is more likely to be room temperature, they're going to be more mold-like or filamentous. It is true they can be in transition, so you may see both yeast and filaments within the same culture, but this should be a defining feature as they switch from body temperature in vivo to growing out in the environment in vitro. The life cycle of the fungi is interesting because they can go back and forth between asexual and sexual reproduction. When you talk about asexual reproduction, this is going to occur in an environment that is very favorable for the growth of the organism. Basically, asexual reproduction, they're just cloning themselves over and over again. So you'll see something like an aerial hyphae going up. In this case, we're looking at something that is making a sporangium, and so this is going to have a thin membrane sac around it. That is designed so that any air currents or movement is going to cause that sac to burst, and then the sporangiospores are dispersed out into the environment. As the spores land on a favorable place to grow, they will start to germinate and vegetative hyphae are going to be formed. However, sometimes the organism needs genetic diversity. That's the purpose of sexual reproduction. So if it needs to get a little bit more diversity in its gene pool, you can see hyphae coming together. In this case, we're talking about the conjugation fungi, and these gametes then form at the tip of the hyphae. They then exchange their cytoplasm and nuclei, and they end up developing a sexual spore. In this case is what's referred to as a zygospore. Kind of looks like a pine cone or a little spiny gumball in between the bulging hyphae. They're then going to be diploid for a short period of time, but then will go through meiosis to get back down to the haploid number, and these are then again put into the sporangiospores. When we look at the sexual reproduction, though, you can see that the aerial hyphae is coming out of the actual zygospore, so this is going to disperse in a similar way as what we see for asexual reproduction. So we see that fungi can go back and forth in between these two modes for reproduction. For the hyphae can be septate or coencytic. With the septate, they have cross walls, and they're a little bit porous, so you can see organelles and nuclei being able to be distributed, but coencytic hyphae actually have no septa at all. There's a lot of debate on whether that is truly a multicellular organism, if there's no cell division there for the most part. But you can see with the coencytic hyphae, the nuclei and other organelles are just free to be able to flow from one portion of the organism to another. In the other picture to the left-hand bottom, you see an example of asexual spores being formed. These are going to be produced by mitosis and subsequent cell division, and fungi are very prolific in producing their spores, and they literally get all over in the environment. They are very easily dispersed. For the type of asexual spores, in the lab this week, we'll focus mostly on the conidiospores and the sporangiospores. The conidiospores are the ones in panel A. They're formed in chains, so you can see that at the top of the conidiophore, they're going to be the chains that are referred to as the conidia. For the sporangiospores, they're in panel E. The sporangiospores are a sac, It's hard to see the membrane around all those spores, but again, both of these are designed for quick dispersal. Any current, any movement is going to disperse these into the environment quite readily. We also have in panel B the arthrospores. They are simply for the long filaments of fungi, the fragmentation or the breaking off of these now arthrospores. In panel C, this is what happens often in the yeast for budding. You see the pseudohyphae, and they are then budding off what is referred to as a blastoconidia. We can also in panel D see an enlargement within the hyphae. These are referred to as chlamydoconidium, and it's just literally a bulge where it will start to then break off and form a new thallus as it finds some nutritional source to be able to grow. So it is more common to see the asexual spores, but it's going to be the sexual spores that we use for classification. So sexual reproduction occurs in 3 phases. Plasmogamy is going to be when you have a haploid donor cell, which is going to be labeled as positive, penetrates into the cytoplasm of the recipient cell. Chirogamy means that you have the fusion of the 2 nuclei. This is going to form a diploid zygote, and then it has to get back down to the haploid. It'll undergo meiosis so that this will then produce the haploid nuclei, and these are going to then be housed within the sexual spores. The purpose of sexual reproduction is that some of these are now going to be genetic recombinants. Again, diversifying the gene pool means that the fungi may be able to survive in a new or an adverse environment, whereas in asexual reproduction, conditions are perfect so that the organism just keeps on cloning itself over and over again. So looking at the life cycles again, asexual reproduction meant to just clone, get lots of those spores out into the environment. The purpose of sexual reproduction is to get some genetic recombination, even though you are effectively putting spores out into the environment in a similar way. They're going to have genetic diversity that is going to hopefully allow them, under selective pressures, to survive. The type of sexual spores that we see are defining the phyla. So in the top panel, we see the zygospore, which was in the life cycle previously. This is going to be the fusion again of the gametes at the bulging end of the hyphae, and you get that kind of gumball, spiny-looking spore in between. The ascospores are also formed in a sac. So this is referred to as an ascus. And so we see here that there are fewer spores because reproduction often is costly. And so we see a sac with these spores inside. The basidiospores are mostly found in things like mushrooms. They have a basidium or a pedestal, and then the basidiospores are hanging down off of the basidium, that pedestal. As I said, the medically important phyla of fungi are defined by the type of sexual spore that they have. That doesn't mean that they do not have asexual spores. It means that they have both. So the phyla that are named here in red are all telomorphs. That means that they produce both sexual and asexual spores. So we have the zygomycota, which are the conjugation fungi. Those 2 hyphae coming together, so they make the sexual zygospores. But when they do have asexual reproduction, it is by sporangiospores. The ascomycota are the sac fungi. So their sexual spores, the ascospores, are formed within a sac or an ascus. Again, they do have asexual spores. These, unfortunately, are not in a sac. These are in chains. These are the conidiospores. So make sure you realize that there are some of the sac fungi that are the true sac fungi for their sexual ascospores, but we also have the sporangiospores that are in a sac, so don't confuse the two. We also have some fungi that have lost the ability to reproduce sexually. These are referred to as the anomorphs. We believe that they lost their ability to sexually reproduce because they became well adapted to their environment. There had been a theory that they just didn't have sexual reproduction. However, we believe that is no longer the case, and we believe the Animorphs actually came from the Ascomycota. The Basidiomycota are referred to as the club fungi. That basidium kind of looks like a club hanging down from the bottom gills of the mushroom. So the basidiospores are the sexual spores. Again, they also have asexual spores. In this case, again, the conidiospores, which are in chains. So historically, you may see another name that is not used anymore, but if we could not find the sexual cycle, then we put everything into a category that was referred to as the Deuteromycota. Now that we have modern techniques where we can classify things by their ribosomal RNA, we have been able to appropriately place things into their appropriate phyla, even though we may not observe their sexual form. We're going to talk much later in other chapters about the fungal diseases or the mycoses, but just to give you a few hints of what these types of diseases are, they are often the cutaneous and or superficial mycoses, so things that affect skin and nails and hair. Every once in a while, subcutaneous, they get under the skin, but we also see some issues with more systemic mycoses as well, especially as they may come in through the respiratory route due to inhalation of the spores. In general, though, the fungi are not outright pathogenic. They're mostly opportunistic pathogens that when normal flora are reduced or people are immunocompromised, that they can then start to establish disease. The Zygomycota, a few examples of the conjugation fungi, are more going to be the environmental ones that you commonly see. So Rhizopus is the black bread mold, gets all over. Mucor we see as a common food contaminant. It's ubiquitous, meaning that it's very common, found almost everywhere, but it can be an opportunistic pathogen, especially causing invasive pulmonary disease as spores are inhaled. The Ascomycota are a very diverse group. The sac fungi can be anything from the cutaneous diseases, such as things like ringworm and athlete's foot, caused by Microsporum and the Trichophyton. So you can see the cracking and scaling of athlete's foot, and ringworm, again, because these grow out in concentric circle, you can see that the middle portion of the lesion has kind of not died out, but it's not as prominent. But as the hyphae keep extending, the ring keeps on getting larger, and it is very inflamed at the edges. Other Ascomycota, the dimorphic Ascomycota are ones that are often regional pathogens. So the Blastomyces dermatitidis, we'll talk about this as being very common down in the states along the Mississippi River. Histoplasma capsulatum, again, overlaps in that it is found in both the Ohio and Mississippi River Valley. Both of these can cause nasty pulmonary infections, but with the Blastomyces dermatitidis, as the name implies, it's also going to have an associated rash. Aspergillus is a common plant and animal pathogen. Again, it is ubiquitous. It is found just about everywhere. You see in the picture here, it's growing on corn. But think about all those spores being put out in the environment. So aspergillosis is a very nasty fungal respiratory disease. But again, you wouldn't expect young, healthy individuals to have such a disease. Most of the time, it occurs in people that either have predisposing conditions such as lung conditions, or they are immunocompromised. The Basidiomycota are the club fungi. These are typically the mushroom. We see here the Amanita mushroom that has the nice red warning cap because it produces a potent neurotoxin. However, it's also harvested for its anti-tumor properties, and so fungi can give us not only a variety of toxins, we also see that they have some very protective properties as well. The member of the Basidiomycota that doesn't seem to fit here is Cryptococcus neoformans. This is an organism that can cause a fungal meningitis, and it's associated with soil and also bird droppings. So we get it by inhalation, and then it is able to cause a meningitis because it is one that has a very large capsule, as you can see in the right-hand panel. People have tried to rename this as the Phyllobacidiella, but you will see people mostly referring to this truly as Cryptococcus, and we'll talk about it later in our central nervous system chapter as one of the leading causes of meningitis. The anamorphs, the best known anamorph is probably Penicillium. It is known for its antibiotic producing properties, but it's a real major agricultural problem for citrus fruits. I'm sure you've seen this if you have a bowl of citrus sitting around and all of a sudden you pick one up and you see the fungus growing on it. More than likely, it's probably the Penicillium. You will note it from its nice green color as the colony, the fungi grow. We also see things in the subcutaneous variety. The Sporothrix is a soil fungi that is notable because it gets into puncture wounds. People have referred to this as the rose gardener's disease because as you prick your finger on a thorn, you can be susceptible to getting these types of spores into that puncture wound. When we have water damage, we often have things like Stachybotrys growing. You see here the water-damaged walls. These spores can be toxic and cause a fatal pulmonary hemorrhage, especially in infants, as they are very prolific, and it's very hard to get rid of the molds. When you do have natural disasters associated with flooding, it's not always the water damage that is the problem, it's the mold that starts to occur afterwards that makes buildings uninhabitable. Other anamorphs that you might know, the Coccidioides is common out in California, and it's a disease that we'll talk about because it looks very much like tuberculosis. Pneumocystis, people know from the standpoint that this is normal flora, but it's associated with the pneumonia that is often found in people that are HIV positive and convert over to AIDS. It means that the pathogen is now the normal flora is not being kept in check, and it becomes a major opportunistic pathogen that often is deadly. Candida albicans, although normal human flora, is often considered the pathogenic yeast. It can cause cutaneous, systemic, or mucocutaneous disease. We see this commonly as thrush. Here you see a pasty appearance to the tongue as the oral thrush gives that kind of bready smell to people's breath. We also see yeast infections in the vagina. Newborns, because they do not have normal flora established yet, they can get it both as the oral form or as diaper rash. And as you might expect, AIDS patients, people are immunosuppressed, and people that are on broad-spectrum antibiotics are going to be more susceptible to these types of yeast infections. There's another fungi that kind of is the other guy. We don't usually talk about the Microspora, which have now been called the Microsporidia, because we've had a hard time classifying whether it's a protozoa or a fungi. We now have classified it as a fungi. This is emerging pathogens such as Nosema and the Encephalotozoan. They're unusual because they do not have a mitochondria. They are non-motile and do not have microtubules, no sexual reproduction, but all of these are intracellular parasites. We first started to notice these in the 1980s. Again, no surprise that this is the era of the beginning of the AIDS epidemic. And these are very interesting pathogens in their life cycle. If you look at the life cycle here of the Microsporidian, you see it looks like a yeast cell almost making a germ tube, but this is going to penetrate into the cell and disperse its cytoplasm and nuclei into that cell. It is then going to grow and reproduce its nuclei and put those then into spores, which are then going to be released, and those can then be put out into the environment. These spores are then ingested or inhaled, and the life cycle continues. The take-home message for fungi is that they are opportunistic pathogens. They are around us all the time. Some of them are our normal flora, but they're mostly harmless in their normal habitat, whether it be on us, in us, or out in the environment. But they become pathogenic in a host who's debilitated. This can be anything from trauma, sometimes stress, or people especially taking broad-spectrum antibiotics that wipe out the bacterial normal flora and let the yeast proliferate. People that are immunosuppressed, either by disease or therapy, are going to be more susceptible. So again, people that have AIDS are going to be very susceptible to these types of opportunistic pathogens. And again, because of the inhalation of spores, people that have predisposing lung conditions are also going to be among those most susceptible to fungal opportunistic diseases. Lest I make you think that all fungi are bad, here's just a reminder of some of the economic good effects from the fungi. Saccharomyces, we know, is one of the major fermenters used for making beers and wines and breads. And this is going to be a good fungi that in order to get it to ferment, we have to make sure that it's in an anaerobic environment. Trichoderma is used for making juices and fabric. And we see things like the Taxomyces is the fungi that is able to produce Taxol, a cancer therapy. That is a very potent production. Again, we saw with some of the other mushrooms the fact that they do have anti-tumor properties. Another important aspect of using fungi is often to control some of our pests. So we see fungi being used to control things like gypsy moth and to kill termites. Instead of using pesticides, these types of natural controls have become popular. The table here is just a list of some of the diseases that are characteristic from the various phyla that we just went over. We'll be talking about these in subsequent chapters, so it's just kind of a highlight of what's to come. Make sure that you go through and check what you need to know about the fungi, what type of spores they have, both asexual and sexual, what are the phyla, again based on their sexual spores, and what are the benefits and the harm from fungi.